So we had a really successful round one, and I think our scholars had a great professional expo. So thank you all for joining us. I know we have some folks coming in for the first time that weren't able to join us this morning. So this is the second part of the Astronaut Scholar Technical Conference. We had 16 great presentations this morning, and so we're up for a few more. I'm going to welcome back our moderators, Carol Fitzgerald Tyler and Ashley Monahan. They will keep us on time. They don't let me do that part. And with that, we'll get started, and thank you all for joining.
Hello. Hello. It's afternoon. Are we awake?
Yes.
Yes. One long time ago, 250 years ago, when I was on a bus tour in Europe for my choir, the choir person that always came on the bus said, "Good morning, you are awake, yes? Good afternoon, you are awake, yes? Hello, you are awake, yes? Thank you." All right, well, we had such a great session this morning. Thank you to all of you who presented. And I know we're going to have an equally wonderful session this afternoon. So super excited. Y'all are going to change the world, and we're very excited to see it. We're here for it, as the kids say. Right? Is that right? So this morning, we talked about a lot of things, including promiscuous voles. We talked about rat cohorts. And we talked about many other things that I was trying to relate to my husband in a call just now, and he said, "Please go back to that and stop talking to me." However, we're very excited for this afternoon. I'm going to turn this over to Ashley. She's going to give you a couple of logistics, and we'll probably end up taking a little bit of a stretch break in the between, and we'll see what happens. Again, my name's Carol Fitzgerald Tyler. In my real life, I run global transformations for big companies that annoy me. But in my personal life, I am on the board of trustees and love working with all of you and helping with your professional development, and you're fantastic. So thanks for being here this afternoon. I'm going to let Ashley do her logistics, and then away we will go.
Okay, friendly reminder for those who did not present this morning. So again, clicker, point it towards the back corner. Okay, you will have a laser pointer, so point at your slides, do whatever you need to do. There's a screen in front of you to where you can see what you're doing. Once again, we will be cutting all off, so please don't make me do that, but if you do, it's okay. This is a safe space. A lot of y'all haven't gotten to present before, so this is an opportunity. It's not meant to be a challenge, okay? So also, I encourage all of you to ask questions. Ask questions of your fellow scholars. If you're a supporter who's here with us, please feel free to ask questions. I'm a firm believer that I don't think there's any such thing as a stupid question, especially in this smart of a crowd. Okay, y'all are talking about really incredible things. So let's ask questions. Let's stay engaged. Okay, I know that post-lunch tiredness is probably hitting a lot of you. A lot of y'all got up really, really early yesterday and early again this morning. So let's keep the energy up. Okay, how does that sound? Okay, let's get started.
All right, everybody, let's welcome. So our first three presenters are actually remote, so they'll be joining us on the big screen over here. So we're going to start with Alicia Chulani. We've talked about voles, worms, and other things. Now we're going to talk about fish robots. So welcome, Alicia, Fish Robot Interaction Modeling.
Are people able to hear me now?
We can hear you. Go ahead.
Awesome. Okay, so hi everyone. My name's Alicia. I'm an engineering student at Harvey Mudd College with the Lab for Autonomous and Intelligent Robotics. And today I'm going to be presenting some of our research on fish-robot interaction modeling. So first, just to provide some context into the problem we're trying to solve, biologists are currently working to study patterns in a wide variety of fish species in an effort to better understand how behavior is modulated by neurobiology. However, we currently don't really have a way to control live fish to exhibit certain behaviors with the goal of observing the response of other fish, and this limits the options to create experimental situations that can accurately study fish interactions. So today I'm going to be presenting some of our work on a robotic testbed, and this is a tool that aims to use bio-inspired robotics to study animal interactions. A main concept of bio-inspired robotics is that not only can these robots use classic closed-loop control, where robots respond to position information, but robots can also use animal-in-the-loop control, where robots base their movement and patterns off of the location of the animals around them and their behavior. Implementing animal-in-the-loop control, our hope is that this will allow this testbed to be a non-invasive way to conduct biological studies with robots that actually respond to live actions of other fish. So moving on into the actual design of the robotic testbed, this testbed consists of 2 levels, an upper tank with fish and the fish model, and a platform underneath where a robot drives and magnetically drags the fish model that's in the upper fish tank. The robot that drives on the platform underneath is mounted with the microbit controller, and this can communicate over radio with a second microbit that's connected to a laptop outside of this test setup, and that runs the software that we use for image processing and trajectory tracking. A video camera is attached to the top of the testbed, and that provides a top-down view of the tank. Individual frames from this video feed can be processed to estimate robot and fish states and things like their XY position and yaw angle. So we can use these frames to do 2 things. First, the robot's going to use the images and decide whether it's going to exhibit a certain behavior for a desired robot-fish interaction. If not, it's then going to just use the videos as a feedback signal for the robot trajectory tracking algorithm. Based on a set of time-dependent points such as x, y, and theta, the trajectory tracking algorithm calculates translational and angular velocities, which are then transmitted from the laptop computer back to the robot via a radio signal. So that's the basic setup for the robotic testbed. We're currently in the process of developing behavior suites that are of interest to biologists. And we started with a wander, detect, evade behavior, which is currently being used to identify predatory behaviors of fish. The robot can begin with the wander motion, where it's traveling in a rectangular path across the edges of the tank. Once it reaches a corner, the robot will detect whether it's in a threshold distance from a live fish. If there's a detection that occurs, the robot's then going to evade and take a diagonal path across the tank at a much higher speed. And from this setup, we're looking to observe fish in the corners that are then chasing after the robot, and we can collect video data of this to observe the biomechanics of this interaction. The software that we've developed is also equipped to plot data for desired robot positions, actual robot positions, and the fish positions, as well as robot and fish velocities. So on the right, I'm showing a plot of a top-down view of the desired and actual robot positions, as well as the fish positions overlaid onto that. As you can see, the robot fish is in the middle of an evasion path in this plot. It was able to recognize that there's a fish in the bottom left-hand corner and then evades it, and we can actually see that the fish is following the robot. Currently, this behavior suite is being used in studies that are in partnership with a bunch of neurobiologists at the Keck Science Center, and they have a specific interest on the Burton's mouth brooder species, which portrays a lot of interesting predatorial behavior. Our hope with this tool is to continue to equip it further with plenty of test suites so it can be used by biologists for many different use cases across the country. And we're continuing to work with Keck to look to improve the mechanics and trajectory tracking for the robot as it acts in the water. We're also looking to improve this tool with more complex experiments that use reinforcement learning techniques, and we also want to optimize fish tracking accuracy using an AI model. But yeah, thanks for listening. I'm really grateful for the ASF for allowing us this to also happen remotely, and I just want to give a quick shout out to all my mentors and advisors as well. I'm happy to take questions if anyone has any as well.
Thank you so much, Alicia. Who has a question for Alicia? Here we go.
Hi, I thought your project was very interesting to start with, but I was just wondering what the implementation would be of this. I know that you're modeling the fish and you're tracking the fish and you're taking videos of it, but what kind of application does that have, or what kind of information can we learn from those videos?
So we're hoping that this can kind of be taken to like a bunch of different biology labs across the country. We're using this right now in Southern California with some labs in Claremont, which is where I go to school, as well as the greater LA area. Different biologists have different like desires from this. For example, the people that we're working with are looking specifically at like the biomechanics of the way that fish tails like move when they're chasing after like this robotic fish. Other people have been talking to us to like kind of explore the way that colors change of the fish as well as they're chasing after the robot. We're also looking to like explore how we can change like, for example, I have this algorithm that does specifically for predation, but like for things like territory as well, we're looking to figure out how we can code the robot to do other things as well. So a lot of different applications. Again, this is more of like a tool that we're then going to partner with biologists to like figure out what they're interested in as well. But we just wanted to build a tool that's not as invasive to use to study animals, specifically marine animals.
Great, thank you. Any other questions? One more.
Hello. I was wondering about why was there a difference between the expected position versus the actual position of the robot?
Yeah, so because I'm gonna go back to the images. So because we have like a bunch of electronics on this little robot here, we're not really at the point where this actual robot can be in the water with the tank. So right now, all of the image processing is supporting this robot that's driving underneath the tank. And because this is like a magnet that's then connected to the lure that's in the water, it kind of like as it's traveling and trying to follow the path, this like shakes around a little bit, which is why you're seeing like the shakes here. It's because the lure is like tiny and can't really withstand following directly the path in the water. So that's one of the things that we're looking to improve in future iterations, trying to figure out how we can have it exactly follow the path. But yeah.
Gotcha. Thank you so much.
Thank you, Alicia. We appreciate you joining us. Our next presenter is Santiago Lopez. He is also joining us from remote on C60 laser desorption optimization. Santiago, are you with us?
Yes, I am.
Great, welcome, and go ahead.
Okay. Can you see my screen?
We can see your screen, and you are not on
Okay.
mute.
Okay. So hi, everyone. Today I'm going to present to you some of the work I was doing over the summer at JOA in Professor Jun Yi's lab. It's titled C60 Thin Film Targets for Laser Desorption. So the goal of this project is to be able to produce targets that we will shoot lasers at and then study the molecules that come off of these targets. And so the molecules that we're studying are C60. So first I want to give you a broad overview of why we want to study molecules. So recently, a lot of work has been going into actually studying individual atoms, being able to control them using really precise lasers and optical setups. And if we're able to control the state of these atoms, the position, or the interactions between them, we can do some really interesting stuff, some of which includes doing fundamental physics and chemistry, even making computers out of them. On the other hand, molecules, there's not much work being done on them yet. There's only a few small molecules which people have been able to prove to be useful so far, just because they are so complex. So just because of all the complexities of the molecules and how many atoms are in them, it's very difficult to control the individual states of these molecules. So in order to be able to start to do that stuff, we have to really characterize the molecules and find what frequencies excite them and control them well. So here's just a few papers I found, some reviews, just showing that people are looking into studying molecules, and they do have a lot of worth, and there's a lot of interesting things we can do with them. So why do we want to study C60? So this is a very cool molecule. First of all, it's shaped exactly like a soccer ball, which I thought was pretty cool when I first saw it. But basically, it's composed of 60 carbon atoms, and they're all in this sort of soccer ball configuration, and it's called a buckyball. And so there's a lot of symmetries that this molecule has and special properties that make it really cool to study and basically make it much easier to study as well. I'm not going to go into too much of the details, but if you have questions about that, you can certainly ask. And so the way that we study this molecule is by using something called spectroscopy. So this is sort of a simplified version of what we do. So you can see that there's this circular target in the back, and we shoot this high-powered green laser at it. And that is the desorption laser. So that is what takes the solid molecule that is on this target and extracts it and makes it into a gas. Then this gas is floating around, and we have another laser, which is the red one, and this laser is performing spectroscopy on these molecules. So what's happening during spectroscopy is basically that you have this energy coming in And if it is at the right frequency, then these molecules will absorb this energy. And so we want to see what frequencies these molecules absorb energy, because that is how you're able to see how you're able to control them eventually using these specific frequencies. So this is just a sort of simplified model of this. And again, if you have questions on this, you can definitely ask after. So what did we do? So before, to be able to produce these targets, there was a method called thermal evaporation. This method was pretty inefficient and also took a lot of time. So what they did was basically have a metal boat with C60 powder at the bottom, and they would heat it up using electrodes, and then the C60 would evaporate up onto this tantalum target. You would get a really uniform layer, and it was really good. However, you lost a lot of C60 powder. It would just go kind of everywhere, as you can see kind of in this cone shape. And then it also took like 5 to 6 hours to do. And nobody really wants to be there for 5 to 6 hours doing this. So we wanted to come up with a method to be able to produce these targets that was quicker and also didn't lose as much C60, because C60 is extremely expensive. And so we came up with a method called spray coating. What we did was get a airbrush, and some people may have seen, it's kind of like an artist airbrush that they use to make like cool designs and stuff. So we got one of those, and then we hooked it up with air lines and then a C60 toluene solution, and we would spray it down onto a target. The target was heated up so that the toluene would evaporate really quickly, and then at the end you would just get a thin film of C60 left on this target. This process was a lot quicker than the other one. I think the spraying took about 2 to 3 minutes. And then also any C60 that was lost was actually just lost on this copper heat sink that was there. So we can just take the heat sink and wash it and get whatever C60 was on there and reuse it even. So we were able to come up with this method. And this is sort of a result of what the uniformity and thicknesses of the target looked like. So the reason that we care about uniformity and thickness is because we want to be able to desorb the same amount of C60 every time that we shoot the green laser at the target. If it's not uniform, we're going to get different amounts each time, and the quality of the spectroscopy will not be that good. So this is just showing that we had a pretty uniform thickness. And then this is the absorption signal that we see from our targets compared to the thermal evaporated targets. So we only had kind of 2 to 3 months to make this method, and there's still many, many things to optimize, but we did see signal, which is good. So you can see here, the blue curve is the spray method that we created versus the green curve is the thermal evaporated method. And so we're still trying to optimize different things to be able to get the signal to noise ratio to improve for our method. And then lastly, I kind of want to just show you the absorption spots that we made. So this is every shot that we shot at the target using the absorption laser. You can see that we're making a pretty deep indent into the C60 and extracting some of it. So this is just kind of a cool picture to show that. But one thing to notice is like we're not desorbing all the way through. So that's kind of one of the things we can optimize. So overall, we were able to develop a new method to shorten the time for producing targets and improve the efficiency yields. Additionally, we showed that, well, additionally, it has to be optimized. That's one thing. And then future work is going to focus on further studying C60 and its different states and being able to control it well in different environments. And so with this, I'd just like to thank the people that I worked with and, of course, ASF for hosting this event. Sadly, I wasn't able to be there in person, but I'm glad I was able to at least join virtually.
We are glad to. Thank you, Santiago. We have time for one question right now.
Hey, thanks so much for the great presentation. So C60 has some pretty cool irreversible point group symmetries because it's part of the icosahedral point group. What does that entail in terms of its vibrational and rotational spectrum? Are there any issues with actually desorbing it because of its unique symmetry?
That's a great question. So actually, its symmetry is why it's useful in some cases, because there's a lot of the different transitions in its rho vibrational spectra that are blocked because of these symmetries. So there's a lot of theory that goes into it, but basically you have sort of irreducible representations of this point group, and certain transitions can be modeled using these irreducibles, and they can only transition to each other if they are composed of the same irreducibles. So that's kind of, if you want to look into that, that's kind of, that answers that question. In terms of the desorption, for now we're just using this green laser just because it's something that was kind of lying around and it works. And we looked at its absorption spectra for that. The desorption is kind of just like, we're trying to just push as much energy into the system as possible to get it off of the target into gaseous form. The spectroscopy is where the symmetries come into play.
Thank you so much. Excellent presentation. So, a quick show of hands. Who has a green laser lying around? Anybody? Okay, good. Thank you. Thanks. We'll talk later. Okay, next, I have to show just a little bit of happiness. Tara Young is joining us from the University of Washington. Tara, I had the pleasure of meeting Tara in March when we went out to visit. She's going to be talking about a DNA binding protein and how it recruits things that I can't pronounce. So Tara, are you there? Yay! Welcome. Go ahead.
Thank you. Thank you so much, everyone, for attending my presentation today. My name is Tara Young, and I am a rising senior at the University of Washington, where I'm a biochemistry student, and I conduct research in the Department of Microbiology in the School of Medicine. Today, I would like to share with you my project on discovering an interaction between two DNA replication proteins and their implication on drugs like chemotherapeutics and antibiotics. So first, when I talk about DNA replication, I want to give you a little bit of an idea of the challenge that it presents in the cell. So first, you can think of your DNA a little bit like the earbuds that get stuck in your pocket and are tangled. If you're to pull them apart, you actually generate a knot that's really hard to resolve. And the same exact thing happens to your DNA because it's being unwound from a state where it's already wrapped around each other. Now, this generates these knots, and we call them positive supercoils. And these positive supercoils prevent further replication and must be resolved in the cell. Now, our cells are luckier than we are. They have enzymes called topoisomerases, which we refer to as these molecular scissors that go in, cleave these knots of DNA, relax them, and then glue them back together so that your DNA is easy to be replicated further. Now, because of this function, topoisomerases are very common drug targets in both antibiotics and chemotherapeutics because they help to prevent cells from dividing and growing any further. Because without a replicated DNA, bacteria and cancer can't divide. And so this is very important, but there's some really big unanswered questions in the field, and that's where my research comes in. So one of the biggest questions that has existed for a while is how are these scissors actually finding these knots of DNA? They're not known to bind to the DNA and slide along it. So there must be some mechanism in which they're actually able to find these. And I am answering this with my research, which studies GAP-R, which is a DNA binding protein that has specific affinity for these positively supercoiled DNA and has also been shown to interact with topoisomerases by us. Now, I want to show you today my really exciting model that I've developed in the past year about how this interaction may occur. But first, I want to give you a little bit of context as to what brought us here. So the first thing, I've been working on this project for 3 years now, which is a very long time to be working on one thing. And we weren't entirely sure what to make of our data. And so I brought this to another lab at the University of Washington, the David Baker Lab, and they work on protein design and prediction. And one of my friends said, well, have you considered what the structure might look like if you put it into a prediction software like AlphaFold? And I said no. And so thankfully, using their resources, they were able to run predictions. And what we found was really exciting. So let me show you first, this is an established structure of a topoisomerase. What you'll notice is in the green, we have the A subunit on the bottom and the B subunit on the top. Now, each time we ran these predictions, we found that my protein GAP-R, which is here in magenta, was always predicted to be in the exact same spot of the topoisomerase enzyme as the B subunit, which confused us because the full active enzyme, the scissors, are only active when A and B are together. Now, I'm going to show you the other data. When we ran this each time with the two different homologs of these topoisomerases in bacteria, GIR-A and PAR-C, which are the A subunits, each time, it always placed GAP-R in the exact same locus of the subunit, the same surface. Now, we wanted to run a negative control as well, because another model could be that perhaps AlphaFold is seeing that this entire enzyme is assembled this way and will just predict any other protein to fill this gap. So we predicted the interaction with a protein known to not interact with these enzymes. What we found is that in the gold here, you can see it was actually localizing, it was generating these predictions, moving it all around the protein. It's not just a nature of AlphaFold to predict this, but that it might be actually consequential for the cell. So really quickly, I want to bring you to our model now. Our model is that gap R is bringing the A subunit to these supercoils. There's an exchange for this B subunit that is competitively interacting. Not both can be there at the same time. And then this reconstitutes these scissors, the topoisomerase, that can relax the supercoil and then falls off the DNA. And then in some step, gap R and the A subunit are associating either on or off the DNA and then going back to the supercoil. So this is our model for how this mechanism could be working. And there's some really important implications to this. So GAPR is the very first discovered topoisomerase recruiter, and it provides a way to separate these scissors. Because you can imagine if you have functional scissors that can cut your DNA, both strands of it, it could be very dangerous and it needs to be highly controlled. And by this mechanism, it's interacting only with one subunit at a time, which helps to protect the cell. And the second is it proposes a new mechanism to inhibit replication by inhibiting the specific interaction surface. Thank you so much for listening to my presentation today. And I would like to acknowledge everyone in the Will Lab and also in the Baker Lab, and especially John and Carol Tyler for funding my astronaut scholarship. Thank you. And I would like to take questions.
Thank you, Tara. Let's take a question.
Thank you. So my lab also uses AlphaFold quite a bit. I was wondering if you compared the versions AlphaFold 1, 2, and 3, and if there was a significant or improvement across the 3 versions.
Yes, that's an amazing question. Yes, so we've run it not with AlphaFold 1, but with AlphaFold 2 and 3. And what we found is each of the models that we got from the predictions were all very consistent in where they placed GAPR. And I know a little bit less about the technical aspects. That's much more on our collaborator who work much closer with this. But on our end of it, we didn't see much difference in the quality or where it was orienting GAPR in these predictions. The one difference, as I'm sure you're aware of if you use AlphaFold, is that AlphaFold 3 can now also have DNA in these predictions. And when we tried predicting the interaction with DNA, because of course we're assuming that this interaction must be in the context of DNA, what we found is it would essentially loop and thread the DNA through the structure wherever it could find a gap, but it always had very low confidence in where it was placing the DNA. So we kind of feel like we're at the maximum that we can get with predictions. And now we are trying to work towards perhaps a real structure of this interaction to verify it.
Great. One more question.
So after identifying the binding via AlphaFold, are there any mutations you can make to either of your proteins to verify this experience in vitro?
Yes, that is such a good question. That was a lot of my project for the first 2 years was actually making different truncations or point mutations. And what I found is that between these predicted interactions is it predicts quite a large surface of interaction, and it appears to be important also how GAPR is folded. We've generated a mutant that doesn't form a tetramer like the box I had been showing you. Instead, it forms like 2 lines, and that no longer interacts. So we have some idea about maybe the shape, but we don't know specific residues yet. And unfortunately, with the level of resolution that AlphaFold provides us, we can't, predict specific like salt bridges or interactions like that. That would be really helpful in figuring out the best residues to mutate. But hopefully in the future, with a verified structure by cryo-EM or X-ray crystallography, we might get closer to figuring out exactly what we can mutate and change.
Thank you.
Thank you so much, Tara. Very good. We wish you were here in person.
I wish so too. Thank you.
And I must say, Tara took her inspiration from my headphones that constantly find ways to knot themselves up while I'm not looking. Thank you very much.
Yeah, mine do too.
Awesome. All right, now let's greet our first in-person presenter for this afternoon, Zoe Winston, a metabolomics analysis of pregnancy complications. Welcome, Zoe.
All right, hello everyone. My name is Zoe Winston. I'm a math major at the United States Military Academy, and this is my presentation on a metabolomics analysis of pregnancy complications. Yes. So, hypertensive disorders of pregnancy, or HDP, is the leading cause of maternal mortality. It affects 16% of pregnancies worldwide and leads to 30,000 deaths on average every single year. As such, it's incredibly important that we increase our understanding of this disease and increase our treatment abilities. So in order to do this, we turn towards biomarkers, right? Biomarkers provide us a unique insight towards diagnosing this disease before the effects become critical. In order to identify these biomarkers, we turn towards the field of metabolomics. All right, now what is metabolomics? Okay, so metabolomics is the study of metabolites, and specifically it's quantifying the different concentrations of metabolites in patients with a disease such as HDP when compared to control patients. This allows us to identify biomarkers to be used in clinical settings, and it informs targeted therapies, because if we know what metabolites and what metabolic pathways are disturbed in patients with a disease, then we can target those in our treatments. In this study, I'm working with data from blood serum samples taken from 160 patients from 3 hospitals across Seattle. From these 160 patients, 51 of them were diagnosed with HDP, and the other 109 of them are control patients. And for each patient, I have data on 3,100 different metabolites. All right, so this is an extremely high-dimensional data set, right? I'm working with 160 patients, and I have data on 3,100 different variables. And this doesn't even include all the survey data they filled out at every hospital visit, all of the information about previous pregnancies, their home situations, et cetera. All right, so we're working with extremely high-dimensional data. So how do we do this? In metabolomics, the industry standard is the OPLSDA, or the Orthogonal Projection to Latent Structures Discriminant Analysis. All right, so this is a mouthful. Essentially, what this statistical test does is similar to a PCA. It works to differentiate between 2 groups. So here we have in red are healthy patients, and in green are at-risk patients, so those who have been diagnosed with HDP. And this test is working to maximize the separation between these 2 groups, essentially. And when it's doing this, it assigns a score to each of the different metabolites, assessing how important that specific metabolite is towards differentiating between the 2 groups. And through this, we can perform a kind of feature selection, right, based off of whether or not that score is significant. However, the problem with this is that it has identified 880 significant metabolites from those 3,100 we started with, all right? Now, this is an absurd number of variables to ask a doctor to measure in an appointment, right? And also think how expensive that doctor's appointment would be. So this was the problem we wanted to address in this study, is how do we decrease the number of significant metabolites that are identified? Now, the other problem I wanted to address with this study is that when I was talking to the biochemists using this and the clinicians using these OPLSDAs, they had no idea what the test was doing, right? They were treating it kind of like this black box problem we see with AI. You put your data in, you get your results out, they're happy, they move on with their lives. But no understanding of what was actually happening under that black box. So in order to address this, I turned towards a battery of statistical analysis with simpler tests such as the stepwise regression, elasto regression, and an elastic net regression, all of which are specifically designed for feature selection. And I was able to narrow down from 880 significant variables to just 15. All right, and in doing this, I also increased the accuracy rate. So this means that 20% less women would be diagnosed with this, with HDP, when in reality they had a perfectly healthy pregnancy. All right, think of the time, the stress, and the money that saves on behalf of these mothers. Now, the problem with going from 880 significant variables to just 15 is that we need to make sure we're still encompassing the complexity of these diseases, right? HDP is a group of diseases, not just one. So what you can see here is metabolic pathways, and this is the complicated relationship between metabolites and the specific pathways they belong to. So we are looking now at the totality of the pathway instead of just the metabolites, and we were able to narrow down from thousands of pathways to just 4 significant ones. And key to note is the bile acids pathway, which has previously been implicated in HDP to show the severity of the disease. So because of this, we already know it's clinically feasible. So if we move up the time in the pregnancy that we are looking to see for disturbances in the bile acids pathway, we would be able to gain a better understanding of whether or not they're going to develop HDP and how severe that case would be, which provides more time for doctors to mitigate these adverse effects. This concludes my presentation, and I would like to thank the NIH Nutrition for Precision Health grant for providing the funding for this study. Thank you.
Thank you, Zoe. Question?
Hi, great presentation. So I am unfortunately a biochemist that works all the time with multi-omic approaches, so I am going to ask a multi-omic question. I thought all your data was really great, and it's amazing that you narrowed it down to just 15 metabolites. For me, my next question would be, what other data can you get to hopefully define a more precise mechanism for what's going on and how you can intervene. For us, we ultimately integrate almost all of our metabolomic data with proteomic data and phosphoproteomic data. So is there any way that you have access to other tissue that you could perhaps do more in-depth analysis on to get a more comprehensive picture, or what are the plans for that in the future?
So unfortunately for this data specifically, we only have access to metabolomics data. So this was taken as a part of the Global Alliance to prevent preterm birth and stillbirth. I think that's what GAPS, GAPPS, is what the data set is from. And from that, we only had blood serum samples for us to do mass spectrometry for metabolomics data. Going forward, as a part of this NIH Nutrition for Precision Health grant also, we're looking to team up between schools that are data centers, such as my school, along with like research hospitals and things like that to get more data on this. But that's a question of money and time on behalf of
Quick
researchers.
follow-up. Are you planning to use the NIH as a All of Us database? Because that just became publicly available.
So that's part of the MPH grant. So we fall under the All of Us project.
Okay, awesome. Thank you so much.
Yes.
Any other questions?
Yes, very interesting talk, thank you. How easy is it to test for those 15 things that you described for a common doctor?
So it would be very easy, but this needs to go through a lot of clinical tests, how all medical data works, right? So this is not like I found these now tomorrow, someone can go get tested for this. The other problem is that the data I'm working with is untargeted metabolomics data, which anyone who's worked with omics data, we haven't identified necessarily all metabolites that we can find in data. So essentially how untargeted works is we say, okay, what is in this blood sample? Literally anything. We might not know what that is and what specific function it has, but we've identified it as being present in that sample. So of the 15 I identified, only 13 of them have actually been annotated, and do we actually know what specific metabolite that is? So that's another roadblock we have, but the lab I'm working with at University of North Carolina, Chapel Hill, they have a Nutrition Research Institute where they have people specifically dedicated towards identifying those metabolites. So this is still a relatively new field in biochemistry, so we're working on that.
Yes. Thank you so much, Zoe. Fascinating. Our next presenter is Jane Santamore. She's going to be speaking on the stability of dynamics between melanoma cells and an immunotherapy-induced response. Welcome, Jane.
Good afternoon, everyone. My name is Jane Santamore, and I am currently a junior at the United States Air Force Academy, and today I'm going to be talking about the stability of dynamics between melanoma cells and an immunotherapy-induced response. So this is a quick overview. And then starting with the introduction, I feel like we all hear about melanomas a lot because melanomas are actually one of the most common cancers for young adults. A common therapy for melanomas is called immunotherapy, which is essentially harnessing your body's immune response to treat melanomas. And in this study, I looked at data from Marzio Panisi, who looked at melanomas in lab mice and essentially developed a system of differential equations to kind of model the rates between melanomas and all of the interactions in their bodies. And I did a lot of analysis with MATLAB. So this is kind of a flowchart of the biological system. You don't really need to know what all the arrows are or what all the boxes mean. The only thing you really need to take away from this is the fact that trying to model facets of the human body and interactions between cancer and the immune system is incredibly complicated and requires doing all of these accurately. And this figure translates into these differential equations. There's a lot of variables, a lot of parameters. Again, don't really need to focus on the specifics of that, but just know that all of these work together to kind of like accurately model melanomas. And the main equation to focus on is kind of the one in the center here about the melanoma tumor cells. So these are all the variables and parameters, their explanations, again, just to prove that there's math behind it, but we don't really need to know those. So there's different ways to model the growth of cancer. I'm going to kind of skim through these, but the first one is Gompertz, highlighted in green, and then exponential, which I feel like we've all heard of, also highlighted in green, and logistic growth. And then that produces all three of these charts. And essentially, this is kind of helpful to show that cancer, at least in this scenario, will decrease using this green immune response with immunotherapy, regardless of how you model it. And then next up, we can investigate some of the parameters within the system. So essentially showing when you change the parameters deliberately, how that affects all of your variables. And this one makes a lot of intuitive sense because as you're increasing alpha 3, which is our max killing rate of the cancer. So as we can kill more cancer, the cancer dies more quickly, as seen in this blue line at the bottom here. And if we extend the time at the bottom here to 4 months, the cancer continues to go down. Now, we can take this and do more analysis called sensitivity analysis. So basically that's saying when we change our parameters, which variables are the most affected. So when we're increasing the rate, alpha 3, at which the cancer is killed, which variables are impacted the most. So once again, a lot of math that goes into this. Whoops. Highlighted here, but again, that doesn't really matter as much as the graphs do. So this is our graph result, and the main thing to take away from this is this horizontal line here. That is everything unaffected really by changes in the killing rate. And there's one line that's going down kind of at the center there, and that is some of our antibody parameters, and that basically shows certain variables and certain interactions within the immune system are more impacted by changes in parameters. So essentially, this is just looking at one variable, one parameter, but you can do this for all of the 18 parameters and even more variables that are highlighted in the original system to get a more complex idea of how the human body really interacts and tries to fight cancer. So some future research goals, basically to continue playing with the model and kind of see if we change. This is at the end of the cancer growth equation. So that would be the death parameter for how quickly the cancer cells are being killed. If you kind of mess with that, how that changes the model, and to kind of do more in-depth research about maybe updating the model or fixing some of the facets of it to model cancer more accurately. So then these are my references, and I would like to thank my advisor, Dr. Hilare from the Air Force Academy, and for ASF for allowing me to present. Thank you.
Wonderful, thank you. All right, we have a question.
Alrighty, great talk.
Thank
You looked at like correlations between your parameters and those ones that like the model wasn't like super sensitive to? Could you possibly take those out to simplify your model?
That is true. So we haven't looked at correlations yet. There are a couple variables, or parameters in there that we changed the values of just to make the analysis run a lot faster in MATLAB, but that definitely is something to look forward to in the future. We could do that.
Great, thank you.
One more question.
Hi, that was an amazing talk. Did you look at the difference between cell-based immunotherapies and molecule-based immunotherapies? And if so, how did that change the parameters?
So we didn't have the luxury of that. The kind of data that we pulled from was done in a study using like OT1 and cytotoxic T lymphocytes. We didn't really have the luxury of playing with the kind of immunotherapy that was used. But hopefully if we have more access to resources in the future, that is definitely something that we could kind of test the differences between. Mm-hmm.
Thank you again, Jane. We would next like to welcome Kevin Lee, speaking about the special values of Bessel functions. Welcome, Kevin.
Hello, my name is Kevin Lee. I'm a senior mathematics major at Texas A&M University. This past summer, I was fortunate enough to work with David Cates under Dr. Elad Zellinger at the University of Michigan Math REU. So my talk is titled Special Values of the Bessel Function, and by the end of the talk, I'm hoping that you'll get an idea of what pure mathematics research looks like. Okay, so why do we care and what is the problem? So the local Langlands correspondence is a really important conjecture in modern number theory, but it works over local fields, and local fields are really hard because when you take integrals, you have to prove that the integrals converge before you can even work with them. But finite fields are nice because the integrals are just finite sums, and you know those definitely converge. So our project is interested in computing the value of this matrix over the Bessel function. So this is a matrix function that takes the matrices and maps to complex numbers over these matrix groups. So this is the orthogonal group, the symplectic group, and the unitary group over a finite field FQ. So these are subgroups of the GLN. You can think of these as invertible matrices with extra properties. The hope is that the final formula we obtain can be presented in one theorem because the matrices all have some similar structure. And we are extending a method that works in the local fields case to the finite fields case. Let's talk about some of the main characters in our play. So informally, groups are just sets with operations that obey certain properties, and fields are additive groups with an additional multiplicative structure. So for example, you can think of real numbers with addition, so you can add two numbers together. For a field, you can think of the complex numbers. So these are the imaginary numbers with addition and multiplication. And if you've ever done modular arithmetic, that's an example of a finite field for a prime. In general, though, groups are really hard to understand, so we often view them through the lens of what's called representation theory. Representation theory takes our group and maps it to GLN-C, so these are invertible linear operators on C vector spaces, or just matrices that are invertible with entries in C. And this is nice because linear algebra has a lot of structure that we already know how to do, and we can apply the methods that we know from this field. So a character is a one-dimensional representation from G to C, so that just means it's a function from G to the complex numbers, and usually the group will be FQ or FQ minus 0. All right, so we're going to talk about the specifics of representations of matrix groups. So we want to define a character on this subgroup. So these are the diagonal upper triangular matrices with ones on the diagonal and 0 below the diagonal and whatever we want above the diagonal. So for a fixed additive character, C, we can define a character by essentially summing over half of this additive character evaluated on the superdiagonal, so half of the things above the superdiagonal. And matrix representations are nice because we can construct representations by considering smaller matrix groups and combining these representations through what's called parabolic induction. And that's just denoted by pi circ t, tau. In this case, our elements can be identified with functions from G to C with some additional properties under the group action. So our main character, the Bessel function, is a unique function satisfying the fact that it's 1 on the identity matrix, and it satisfies this right multiplication property. Finally, we say a representation is of Whittaker type if there's a unique vector up to scalar that satisfies, again, some multiplication property. We're ready to describe the method. So for a representation, we can define what's called an intertwining operator, and this takes us from one representation to another. But remember, the Whittaker vectors are unique up to a scalar, so we can unambiguously define this to be the gamma factor. Using the definition of the intertwining operator, we can write our gamma factor as a sum over the Bessel function, so in the orthogonal group case, we get something that looks like this. And then finally, we can apply the Mellin transform, which you can think of as a finite Fourier transform to isolate the value of the Bessel function that we want. So in the symplectic group case, we get something like this. We can recursively apply this definition to get an explicit formula. It was just too long to fit on these slides. So we're still finishing the computation for the unitary and orthogonal group case, but the proofs have been really similar so far. And future projects could compute different values of the Bessel function with, say, larger blocks in these entries. So n by n instead of just 1 by 1. And thanks. I'd like to thank the University of Michigan for sponsoring me this summer and my mentor there. And I'd also like to thank the ASF for having us all here.
Thank you, Kevin. All right. We have a question right away.
How do you conduct your research? Is it just sitting there with a piece of paper and pen, or use computers?
So, I mean, the main thing is just theorems, so we have to think about how to prove the theorems. But a big important thing was decomposing matrices. So this multiplication property, we want like this matrix to appear. So we would take a matrix and decompose it into a product of 3 matrices, and you could use some code to get a guess for what the decomposition looks like. So I use Sage Math, which is based on Python.
Gotcha.
One more question.
Thank you for the talk. So I had a quick question. So you were considering finite fields.
Mm-hmm.
When you're doing these representations, you're only getting like the additive structure. You're not really getting the multiplicative structure. Is that right? Or are you also getting the ring component as well?
No, you do use In this definition, you do use like only an additive character, but the multiplicative characters do pop up. So you'll have just fx. I don't know if that answered your question.
That works. Thank you so much.
Okay.
Thank you, Kevin. And just a quick announcement, because we all love modular arithmetic. There'll be a modular arithmetic water aerobics class tomorrow in the pool at 6 a.m. Luke Kaufman is our next presenter, and he's going to be talking to us about local measurement strategies for multipartite entanglement quantification. That is a mouthful.
Alrighty. Hello, everyone. My name is Luke Kaufman. I'm a rising senior at the University of Colorado Boulder, and today I'll be telling you about my research in the field of quantum computing, specifically on sort of local measurement strategies for quantifying entanglement in systems of many different particles. So to start off, I'll give a little brief on how quantum computing works. So in normal computers, you can think about information being stored in terms of bits, so sort of zeros and ones. Quantum computers have something very similar called quantum bits or qubits. These guys are very similar to classical bits. Instead of having zeros and ones, ups and downs in terms of voltage, they can also explore every possibility in between. And this is sort of what we colloquially describe as superposition. So if you think about a classical bit as being sort of up and down along this z-axis, zeros and ones, a quantum bit has the property to be able to go sort of anywhere in between on the surface of the sphere. And it has a lot of different properties that make it very interesting and allows us to do cool things with quantum mechanics. However, the drawback is that whenever we ask questions about this sort of system, we sort of destroy this property in superposition and get probabilistic outcomes. And what that basically means is that when we look and ask questions, we're going to get some binary information out, zeros or ones, but the probability changes depending on the sort of questions that we ask. Now, entanglement is another very interesting part of quantum mechanics that distinguishes it from classical mechanics. And you can sort of think about quantum entanglement as like non-classical, non-local correlations between quantum systems. And this sort of means that these entangled systems cannot be described solely in terms of parts. So you can't really break them apart. They're sort of intrinsically one unit as a whole. And one way to think about this, sort of cartoonishly, is to imagine you have two coins. If you have two coins that sort of represent your qubits here, you can imagine sort of welding the coins together, which is your sort of entanglement. Whenever you sort of flip these coins, which is sort of putting them into a state of superposition, they're sort of in an ambiguous state, but whenever you measure them, they'll always be correlating their outcomes. So if you flip this sort of weird amalgamation of coins welded together, you sort of always get heads with heads or tails with tails. This is sort of how entanglement works in a lot of ways. But entanglement is very strange in that things aren't just entangled or non-entangled. You can also imagine having varying degrees of entanglement. So instead of having these coins, whenever you flip them, always landing together because they're very rigid, you can also imagine sort of putting a rubber band between the two of them. Whenever you have this rubber band and you flip it in the air, it always has the tendency to sort of want to untwist before landing, but there's still a chance that they'll sort of land in opposite correlated states. So this is sort of a playful way to think about entanglement, but it's a very strange concept that we definitely don't understand fully yet. Now, quantifying multipartite entanglement looks like sort of having a system of a bunch of different particles that are all interentangled, and you want to ask questions about how they're all related to each other and basically put a number and say it is 0.9% entangled. So a way that we do this is by looking at something called purity, and this sort of quantifies how noisy a system looks when you ignore the entanglement with the rest of the system. And so a lot of ways this sort of looks like crosstalk between your system. So if you have entanglement, like another coin that you don't know about, and you sort of ignore it, you're missing part of the whole system, but locally to you, that looks like a form of noise, because you can't really discern whether or not you're being entangled with something else or something else is happening to your system. And ways that we can do this that I'm interested in is calculating sort of the concentratable entanglement. And what this looks like is sort of calculating the purity of every single subsystem and averaging. So if you imagine sort of having a system composed of 3 different atoms or particles or whatever you want, quantum systems, you can imagine calculating the purity of just 1 while ignoring 2 and 3, and then imagine calculating 1 and 2 together and ignoring the entanglement with 3, or sort of imagining computing everything in together. This is what the concentratable entanglement does. It sort of looks at everything as a whole. Now the goal is to find some experimentally viable ways of quantifying this information, and I'll sort of jump into that now. One way is to do local randomized measurements. What you basically do is take your qubit, apply a random rotation, and then measure it. When you do this, you can kind of intuitively think that on average, there's an equal probability being up and down, so the mean looks like 0, but we actually care about the variance. However, this technique experimentally is very hard because you constantly have to sample random rotations in the laboratory. It's very difficult to do. One of my contributions was sort of reducing the complexity of this, so making it a little bit easier to work with. So I basically got this sort of square root improvement in the number of samples that you would have to do in the lab. And this is very difficult because normally with quantum stuff, everything scales exponentially when you try and port it over to classical information. The really cool thing that I'm excited about is this sort of topic called seq POVMs. This is a very strange object, but basically what they allow you to do is go from these exponential number of different measurement settings to just one. And you can still quantify the entanglement using this one single setting. That makes it much easier to do these sort of tasks. However, the issue is that when you want to implement it, it takes additional memory to sort of do it on a quantum hardware. And also, it gives you slightly worse sample complexity. And by sample complexity, I mean number of times you have to prepare a state, measure it, and ask questions about it. But yeah, so in all, these sort of seq POVMs give you a better way to sort of visualize quantum states and ask questions about them, making them more experimentally viable. And knowing these sort of sample complexities, how much work it takes to learn something about a quantum system, gives experimentalists an idea of how difficult a task they're sort of asking and trying to do. In the future, I'd like to find some sort of optimal strategy, which is basically the best you could possibly do, and do things such as finding optimal purity and controllable training measurements. But yeah, thank you so much. I would like to thank Jacob Becky for his support and mentorship over these past couple of years, as well as my PI, Graham Smith. And if you would want, you can also check out the paper here at this QR code if you want. But yeah, thank you so much, and thank you to ASF for having me out here today.
Thank you, Luke. Question for Luke?
Hey, Luke. Thanks for the great talk. My question for you is, how does the viability of your measurement method depend on the Hamiltonian of the system? For example, if I have a more complex Hamiltonian with different properties, do you think it would be harder to use your measurements, your measurement apparatus?
Yeah, that's a really good question. I think usually it depends on sort of what axis you have. So like, to do these types of measurements, like the seq POVMs, what you really care about is having this additional sort of blank information that you can do these measurements on. But like you're saying, if there's this sort of other interactions that are causing it to be difficult, I'm not exactly sure how it would pan out, but I think that's a very interesting question to ask and look forward to.
Thanks.
And one more question.
Hi, I thought your talk was super cool. I'm just curious, what does your lab environment look like?
Yeah, that's a great question. So I think there will be a certain theme going on. I'm a theorist, so a lot of my paper looks like paper and pen work and simulations. But a lot of our group is just sort of working collaboratively and trying to figure things out. It turns into reading a bunch of research papers and learning new math, which is kind of what I enjoy. But yeah, that's sort of the environment.
Super cool, thanks.
Thank you so much.
Thank you so much, Luke. Our next presenter is Luis Santori, and I'm not going to read the whole title because the first part's really cool. Ready? Peering into the moon's past. Come on up.
Hello, everybody. So my project is titled Peering into the Moon's Path, High-Resolution Analysis of Anorthosite Exposures. For anyone that doesn't know, it's hypothesized that the lunar crust is mostly formed of anorthosite, so that's the rock I'm studying. And this work was done alongside Dr. Carrie Donaldson-Hanna and Dr. Melissa Martinot. But for some motivation, the goal is to examine potential differences in the composition of plagioclase within pure anorthosite exposures across the moon. LROC-NAC derived single scattering albedo has been used to distinguish compositional variations at a really high spatial resolution of about 2 meters per pixel. And in this project, we'll focus specifically on computing single scattering albedo for pure anorthosite to gain insights into compositional characteristics. Now, in order to compute single scattering albedo, we start by using NASA's AIM stereo pipeline to create a high resolution DTM, which is just an image which the pixels hold elevation values. Then we take LROC-NAC images and we radiometrically correct them. That helps create accurate reflectance values. We then use SPICE kernels in the DTM to compute ground positions and photometric angles. We then create backplanes for local emission, phase, and incidence, and we map project those images. We map project those images for spatial analysis, and we minimize a simplified version of the Hapke equation to determine optimal values for single scattering albedo. So here's the Hapke photometric model. This is the equation we're minimizing. That table was supposed to have kind of the assumptions we're making and the papers associated with them. But this work was also done by Klett Watkins et al. In 2016, so it's a similar model. Now for single scattering albedo is the ratio of scattering efficiency and total extinction efficiency. Single scattering albedo depends primarily on composition and maturity, and a single scattering albedo greater than or equal to 0.5 are likely areas of pure anorthosite. So in this figure, you have single scattering albedo on the y-axis and mafic content on the x-axis, and as you can see, higher mafic content gives you a lower single scattering albedo. Now for the craters we're studying, we're looking at 11 different craters over the surface of the moon. The color here is for the geochemical terrains that those craters fall under. Now for our analysis, alongside single scattering albedo, we also looked at other parameters and their relationship with single scattering albedo. So that was FeO, so iron content, slope, optical maturity, and rock abundance. For a similar result, single scattering albedo versus FeO, we see a weak negative correlation. We have single scattering albedo in the y-axis and FeO on the x-axis. For optical maturity, we have we have a strong positive correlation with more immature craters having a higher single scattering albedo. And for some conclusions, the differences in observed single scattering albedo are likely not due to inherent differences in pan-plagioclase composition. This supports the standing hypothesis of the lunar magma ocean, which assumes that plagioclase crystallize in a large single event. And the relationships between single scattering albedo, FeO, slope, and optical maturity are likely all related, with areas of high slope less likely to accumulate regolith, giving us higher optical maturity values and lower FeO values. But we need further analysis to understand this interdependence. That's all I have. So here are the references. Thank you.
Thank you, Luis. Do we have a question for Luis? There we go. That front row's got a lot of questions.
Hi. So, correct me if I'm wrong, but it looks like many of the craters that you examined were from the equatorial regions. Is there a reason for that?
So we have some other craters that we're supposed to analyze in the far side. So all the ones we've looked at are in the near side. However, we've had trouble trying to analyze them. So I have like 2 more, and those are more in the. So let's go back to. I think should be. Oh no. No. There's some craters over here that we should be able to analyze soon. So yeah. Does that answer your question?
Yeah, thank you.
One more question for Luis. Anybody
I have
else?
a potential question.
Oh, good.
Are there any future missions to the moon that'll give you more information about this? Or are you looking forward to future space flight to the moon?
Yeah, I am excited. I think, I don't remember the name of the mission. I think it's Lunar Vice that's supposed to go see the Gratisum domes, and we see some anorthosite exposures there. And that hopefully will bring back samples so we can actually see the rock, because you have limitations when you're just looking at a satellite and the images from it. Yep. Thank you.
All right, thank you so much. All right, our next presenter is Margaret Marty. I hope I said that right. Low temperature characterization of dielectric properties of complex oxide heterostructures on silicon. Did I do okay?
Yes.
All right.
Hi everyone, my name is Maggie and I'm currently a senior at Clemson University. Today I'm going to be telling you about my research studying complex oxides at low temperatures. I'll begin by introducing complex oxides, which are a very versatile group of materials with a wide range of physical properties. This versatility stems from the range of energy interactions available in the material systems. An interesting application of complex oxides is quantum transduction, which is necessary for realizing hybrid quantum systems. Particularly, quantum transduction connects superconducting qubits, which operate at 20 millikelvin in the gigahertz range, to optical fibers, which operate at 300 Kelvin in the terahertz range. Now, Transduction can be achieved through electro-optic or optomechanical systems, both of which take advantage of complex oxides. But my research focuses specifically on barium titanate, or BTO. And this is because it's got a particularly high piezoelectric and electro-optic coefficient compared to the more commonly used lithium niobate. Here at the bottom, you can see a transmission electron microscopy image of our samples that we use, and this is just so that you can see the different layers in our material. So we want to look at how these materials and samples behave at cryogenic temperatures because we want to see if there's any phase changes. And the first step in doing this is to fabricate something that we can actually measure. For this, we make metal oxide semiconductor capacitors, or MOSFETs, and our fabrication workflow is shown here. Now, if we want to integrate these materials into on-chip quantum transducers, we need to know how they behave at low temperatures. So we use Gifford-McMahon cryocoolers, or GM cryocoolers. And these are great because they're relatively compact, pretty rapid, and compared to other sources of refrigeration, pretty cheap. So here you can see the inside of one of our cryocoolers, and we mount our sample right here on a custom-designed printed circuit board. So we want to see how low these systems can get. Here we can see a characteristic cool-down curve of our cryocooler where it reached a base temperature of 8.6 Kelvin, which if you don't think in Kelvin, that's negative 265 degrees Celsius. And it did that in about 4 hours. Pretty rapid, you could do a couple samples a day. So after calibrating our system, or after calibrating the cool-down properties of our system, we want to put a sample in it, calibrate it to see how well it's measuring. For this, we used a 100 nanometer thick silicon dioxide sample. We measured the capacitance of our sample using the platform, and then we used the standard parallel plate capacitor formula to calculate the dielectric constant. Here we can see a curve of the dielectric constant as a function of temperature, and we recorded a room temperature value of 3.83, and then we also saw a 36.6% drop in dielectric constant as a function of temperature. After calibrating our system, we were ready to test our barium titanate heterostructures. So there were two primary ones that we were looking at, barium titanate on lanthanum nickel oxide and barium titanate on strontium titanate. So for our BTO on LNO, we saw a room temperature value of 66.2 and a base temperature of 49.7, corresponding to a 24.9% drop in dielectric constant. Similarly, for BTO on STO, we saw a 37.4 value at room temperature and a 24.2 at 10 Kelvin. This once again corresponded to a 35.3% drop in dielectric constant. To conclude, we successfully measured the dielectric properties of barium titanate heterostructures integrated into MOSFETs for the future integration into on-chip quantum transducers. We didn't observe any significant drops in dielectric constant as a function of temperature, and our values were also well below the reported range of 100 to 400, so more research is needed. However, rapid dielectric feedback is still going to be important for future optimization of our growth process. I'd like to thank my teammates at LabEc and our collaborators, and of course the ASF. Thank you.
Thank you, Maggie.
Hi. I understand that perovskites can be pretty tricky to grow. Could you, again, explain maybe a little bit more detail about how these films are actually grown?
Yes, I didn't do any of the fabrication of these samples, but, oh, it's back there. We began with a heavily doped silicon substrate, and then we deposited an oxide on it. There was, depending on the sample, different oxides. And then we used a shadow mask to deposit palladium electrodes on the top that we measured. Like, those were the MOSFETs that we did. But the specifics of the fabrication, I'm not entirely sure on.
Do you have any clue what kind of machine, like ASD, LD, epitaxy was used?
I think it was epitaxy, but I'm not entirely sure.
One more question. Anybody? Oh, there's one.
So I know lithium niobate is sometimes used for like high coherence because it has a higher quality factor. Do you know what the quality factor is of the material here and whether how high quality the resonators you'd make with them are?
I'm not entirely sure. I think that's the next step of our research because this project is being continued at the University of Maryland. So I think we're going to begin developing resonators and then hopefully measuring the quality factor of those once we get there.
All right, thank you, Maggie. So we have seen 9 fantastic presentations, and we are going to not take a break. We're going to take a stretch, and it's going to be led by my esteemed colleague, Ashley, and she's got a surprise for you. She's getting up there to do it. Don't you leave this room. No, stretch time. Do not leave this room.
Okay, so anybody who knows me knows that I love to embarrass myself, so this is going to be one of those times. Okay, I was so inspired by all the presentations. I'm going to start off, be the fish following the robot. Do it with me. I'm not doing this alone. Be the fish following the robot, okay? Okay, then we're going to be the laser. We're going to be the laser. The laser. We're going to be the laser. Okay, what about that albedo? Okay, be the albedo. Okay, metabolomics. Be the circle graph. Be the circle graph. Okay, that low cryogenic temperature. Low temperature. Okay, and then be the math, because I don't know how to be the math. This is the closest I could come up with. I'm not a math theorist. Okay, and then finally, we're excited for those future presentations, right? Okay, that concludes our break.
I'm so invigorated. I love it. All right, you got about 1 minute more to get yourselves resettled, please. 1 minute more. Hello, let's get sorted. Enough. Please have a seat. We have 9 more fabulous presentations to go. Come on, stop being troublemakers. All right, we are going to progress. So I also have the pleasure of announcing another battle of the bands. Not only do we have the promiscuous bowls, but there'll be versus the quantum entanglements on the pool deck later. Thank you for laughing, see? Trying to get it, you got it? All right. She's doing fish, I'm doing the words. Because this doesn't do fish. I have a new hip, we don't fish. All right, let's get started again. Welcome back to the second half of the second technical conference today. We have some amazing presentations yet to come. So we're going to start by welcoming Miguel Mercado, probing topological phases of matter with quantum computers. Welcome, Miguel.
Hi, everyone. My name is Miguel. I'm an aspiring theoretical physicist. And today I want to talk about how you can use quantum computers to answer interesting questions in physics. There we go. So one of the most interesting questions, in my opinion, is trying to understand the behavior of something called a quantum phase of matter. This is where you can get really interesting macroscopic behavior emerge from an individually interacting collection of particles. Emergence is everywhere in nature, from how birds can form these really intricate formations when they're flying in the sky, to how middle schoolers, how boys and girls segregate themselves at middle school dance. It's everywhere in nature. And one of the most interesting types of quantum phases is called the topological phase of matter, which was the subject of the 2016 Nobel Prize in Physics. So this is perhaps one of the most bizarre consequences of emergence, and it was essentially predicted that there exists a class of materials that are invulnerable against environmental perturbations, which is unlike any other material. And to be more specific about this is called topological protection, and it's encoded into something called a topological surface mode. So if you use your imagination for a sec and imagine a box of electrons that are exhibiting this topological property, you can kind of think as this being a quantum memory of the system, that no matter how I deform or distort the structure of the electrons, the physics on the surface will always be preserved. So this is really unique. And some potential applications of this, for example, is in dissipation-less electronic transport, or for example, in tunable quantum sensing. So for example, if the government wanted to build a machine that can detect certain frequencies like uranium underground, being able to control the electronic structure to this capacity could be really useful for that. And this is really great in theory. However, it's really difficult to study these quantum phases in real life. And one promising avenue to do this is what Luke talked about, which is using quantum computers to study them. So a quantum computer is a device specifically designed to exploit the subatomic properties of matter to solve interesting problems in math and physics, using ideas in computer science. So just to briefly illustrate how you can do this to solve the topological phase of matter on a quantum computer, you would first start with an equation called the Hamiltonian in physics, which encodes all the physical properties in a really neat equation. And then you would discretize the time evolution into something called quantum circuits. So for the physicists in the audience, this is like e to the negative I ht over h bar, the time evolution operator. And then you would make these quantum circuits in real life on a real actual quantum computer on a hardware. And so I did this with IBM's quantum superconducting qubits, which is really fun. And then you would perform the measurements at each time step interval, and easy, then you get the dynamics of the Hamiltonian. But that's if we live in a really nice, that's if the world is really nice to us. But unfortunately, we live in a really cold and harsh world, and life is not that easy. And so I want to highlight two things. First, the technology that we're using is just faulty. The materials that we're using is not strong enough to encode the qubits against the environment. And secondly, there's a theoretical problem, namely that the number of circuits required to simulate just a few of these qubits scales exponentially with time. And that is just absolutely despicable. That would mean that the economic cost would do this would just not be viable, and it certainly, our scholarship certainly wouldn't be enough to pay for that. So what our team has done is we've developed a new type of quantum circuit to do this instead for a specific class of systems. So the new apparatus would look like you would start with the Hamiltonian that I mentioned earlier, and then you would turn that into gates, and you can read more about how the quantum circuit works in our recently published paper, or you can read about it for free over on the archive, and then you can get some accurate results from that. And just to prove that it's accurate, we were able to verify our experiment. This is a Fourier transform, the latest experiments on quantum computer, and it agrees pretty well with the theory. And the really cool thing is we were able to actually use the quantum computer to make a new physical discovery, namely that these topological surface modes that I mentioned earlier are robust against the open system evolution in experiments. And this is kind of a non-trivial result because it was predicted that, sure, this could be the case when there's no time evolution in the static picture, but it hasn't exactly been observed in the time evolution yet. And for the layman's, this is essentially just saying that we're corroborating the prediction that was all the rage in 2016. And just to give the main takeaway of my talk, essentially, today's quantum computers could be useful for physicists to study interesting physics. It just requires a little bit more work on our end to make them useful to study the physics. And then finally, I just wanted to thank ASF for this awesome opportunity, and just for the opportunity for me to watch all the talks. It's been great. And I'm happy to take any questions.
Thank you so much. Who has a question?
I have a question that I can start out with.
Okay.
So in your diagrams, it looked like you only had 5 qubits.
That's right.
So you had a 5-qubit line. Are you interested in potentially doing other geometries? And if you had access to something like a neutral atom system that had all-to-all connectivity, how would that change what kind of topological systems you'd investigate?
That's a good question. So that actually is the next direction that we're doing. So we're doing this for larger spin chains. This is just the transverse field Ising model. But we can do this for the XY model and the Heisenberg model for larger systems. I didn't really think about neutral atoms yet, but it is a good question. Exploring this with nearest neighbor interactions would be really interesting, and I don't think that's been done before. So that would be a really interesting project for the future. Yeah.
One more question, please. Anybody?
I have a question. So you said that with a little bit more work, physicists could use this in the field to actually make new discoveries and to improve their research. On a time scale, how soon do you think that might be? Are we talking years, decades?
Yeah, well, it depends how you define impact and what you find is new. If anything, at least we haven't been able to actually image the open dynamics of these topological models yet. So that would be something that's new. Again, it's more of a little contribution. In terms of something more useful, you would probably have to scale this up to just more qubits because all the interesting algorithms that people are studying in theory just require a larger number of qubits. But the good thing is, I think IBM and other companies, and other groups actually here, probably in the room, are working on making machines with larger qubits, which is really cool. Yeah.
Wonderful. Thank you so much. Our next speaker is Josh Ott, and he'll be speaking to us about simulating the critical dynamics of quark gluon plasma.
All right. Hello, everyone. My name is Josh Ott. I'm going to be talking about my work studying the critical dynamics of quark gluon plasma in the physics department at North Carolina State University. So I just want to start off with getting everyone up to speed on what these things are, starting with quarks and gluons. So quarks and gluons are the fundamental particles that make up protons and neutrons, everything around us. They interact with the strong nuclear force, and we describe this force, we understand it through the theory of quantum chromodynamics, or QCD. QCD has a very unique property called asymptotic freedom that says if you have two quarks and you separate them, the force between them gets stronger rather than weaker. So it kind of works like a rubber band, and it means that quarks are kept in confined bound states where we can't see them on their own. If we have a proton and we try to separate a quark from it, nature freaks out and instead makes two new quarks there to make sure everyone has friends nearby to keep energies down. So then if we look at the phase diagram for this matter, you can see at low energies we have hadronic matter where everything is confined. But then if we go to higher energies, you get quark-gluon plasma where everything is free, no longer confined. This corresponds to the beginning of the universe. So microseconds after the Big Bang, everything was quark-gluon plasma. It hadn't resolved into hadrons yet. And here you can see I have this critical endpoint marked down with a question mark because we aren't entirely sure it exists. We know there's a phase transition between these two things, but we're not sure about the critical endpoint. If we want to study it, we have particle colliders. We can smash things together at near the speed of light, but quark-gluon plasma, it only lasts for around 10 to the negative 22 seconds. After that, our little blob of it expands, freezes out, and then resolves into more particles, creating a shower that we can detect. So we don't look at it directly, we can only see what comes from it. And our goal is to study this critical endpoint theoretically to instruct experimentalists on what to look for in their experiments. So our method for doing this comes from universality, where this is a wonderful tool in physics that says we can relate many seemingly unrelated phase transitions together by their dynamics around the critical point. So theorists love this. It means we can take some complex system and instead find a simpler system that's equivalent up to whatever behavior we're interested in, and then study that one instead. So for us, this corresponds to something called Model H, comes from the gas-liquid transition, and this is expected to have the same dynamics as the QCD critical point. So from this, we get our goal, which is to simulate diffusive dynamics, so just diffusion on a conserved order parameter field. It's just a scalar field where the sum over it is conserved over time, coupled to the momentum density of a fluid. So on top of that, we have a vector field, and both of these are interacting with each other, coupled together with differential equations. These pictures, I show the scale invariance of it. So if I zoom in or out on the ordered or disordered phases, you'd be able to tell. But at the critical point, we have domains of all different sizes. It no longer, you couldn't tell if I zoomed in or out on this. So then, actually applying this, we split time steps into three parts: diffusion, noise, and advection, where diffusion and noise can be treated together in what's called the metropolis algorithm, where we have some random flux, Q. Theta there is a random Gaussian variable, and then we propose that as a flux between two adjacent sites. We look at the change in the total energy. If energy goes down, nature likes that, we let it happen. If it doesn't decrease the energy and instead increases it, there's a probability of either accepting or rejecting it. Along with that, we have advection, very similar to Navier-Stokes. We just have to pay very close attention to energy conservation. Here I have some representation of our order parameter in the background with some vectors on top representing our fluid. Actual results from our simulations here, because the Model H had not been simulated numerically before this. On the left, we looked at the renormalized viscosity, where one fun thing theorists get to do is ignore parts of our model that aren't relevant to what we want to study, and then make it easier to work with. So here we have Model H0, which ignores the self-coupling of the momentum field to itself, which allowed us to get lower viscosities observed in the dynamics, which then we used to study the dynamical critical exponent, which is one of the key parameters that characterizes Model H. The goal was to see 3. So z equals 3 was our goal. And for low enough viscosities, we were able to see that, confirming that we had actually simulated this thing. Looking ahead, we want to study cumulants, which are a candidate for an observable, very sensitive to the critical point fluctuations that we can compare with experiment. We also want to study the phase transition in 2 dimensions, because that also has not been studied. Here we've just looked at 3 dimensions, and this method needs to be expanded to stochastic relativistic fluid dynamics, because of course in particle collisions it's highly relativistic. So thank you for your attention. I love to take questions.
Wonderful, thank you. Do we have a quirky question?
You mentioned that when the particles spread out, they begin to shower, and that's how you're able to detect them. What does the showering look like, and how do you detect them?
So in particle collisions, we have these massive detectors, unbelievably large, with many layers of different detectors there. So when we have a collision, you create a bunch of particles because you convert energy into mass, creating particles which decay into other particles, decaying into other particles, and you get a huge shower. It's very overwhelming to look at pictures of it. And then we detect these many, many times a second, so hundreds of thousands of times a second, and then we have to process these in real time and then analyze them. Yeah, very complex process. I don't work with that, I just do the theory, but yeah.
Very cool, thank you.
All right, another question.
So this is definitely something that it's like you need a lot of like brain space to kind of process this, honestly. What do you think like the, I guess, future like as time goes on and you improve your understanding of something like this, what does the relevant like application?
We have no clue.
You're not sure? Okay.
No clue.
That's all. I still think it's really cool. Thank you.
Oh, goodness. Oh, thank you, Josh. And we love the idea that quarks are making friends with other quarks. All right, next up we have Brianna Chen measuring digital micromirror device characteristics for a programmable spectrum source. Welcome, Brianna.
Okay, hi, I'm Brianna, and today I'm going to be talking about measuring digital micromirror device characteristics. So some scientific motivation for this work is that in astrophysics, there are a lot of parameters that we want to measure more accurately. So for example, in type 1a supernova cosmology, you might want to use that to measure the Hubble's constant better, which will inform you about the expansion of the universe. Also in like exoplanet science, you want to know better information about how habitable a planet may be based on its mass, radius, or age. And so to measure these parameters more accurately, you need to have better measurements of flux, so more accurate measurements of flux. And if you can actually reduce the uncertainty in flux from, say, like 2.5% to 0.5%, then you can actually, for example, in exoplanet science, reduce the uncertainty in these parameters by an order of magnitude or more. So it's really important that we reduce systematic uncertainty in flux, and you can do this through calibration, so better calibration methods. So this is the overarching mission that is in design called CANDLE. Essentially, we want to provide sub 0.5% uncertainty in flux through better calibration. And so this is the 0.5% is enough for us to achieve these scientific precision requirements. And so inside CANDLE, there are many different modes, but the mode that is potentially useful is this programmable spectrum mode. So you essentially shape the sun's spectra into some astronomical source that you may want to study the spectra of, and you can send that to telescopes for calibration. So how do you have a programmable spectrum source? So this is actually an instrument at the National Institute for Standards and Technology, otherwise known as NIST, that actually can do this. It's just very large, so you cannot like fly this into space. But you take a spectra, so this is our initial spectrum, and then you can say, I want it to look like this M-type star, for example. So I input this spectra, and I'm like, make it look like this. And then this instrument can actually create light that is of this spectra. And so this is done using this device called a digital micromirror device, or a DMD. And so you can actually see a DMD on the bottom left image. And so that is a pattern that is actually used to create the spectra that is shown right there. So if we look at a DMD, it's essentially about this big, this array of like over 1 million micromirrors like in this small area. And you can turn these mirrors individually on or off at 12 degrees or negative 12 degrees along the diagonal. And so what this is used for is then selecting the intensities and the wavelengths and the corresponding intensities of those wavelengths for CANDLE to shape the spectrum into what you want it to be. So if you zoom in really closely on a DMD, and you zoom even more, you see that there are like a lot of gaps between these micromirrors. So that produces this effect known as diffraction. So essentially, we can model DMDs as a Blaze diffraction grating because they are angled along this thing, and they're also periodic objects. So in particular, this is a Blaise diffraction gradient. So what that means is that we have a specific diffraction order that has maximal efficiency compared to the other diffraction orders, and that corresponds to a specific wavelength. And so there are three things that we really need to notice here, which is first, if we change the distance between the mirrors, or the size of the mirrors, sorry, that changes the properties of diffraction. The second is that diffraction really arises at longer wavelengths, so once we reach the infrared wavelengths, we need to start counting for diffraction more. The third is that we define the diffraction efficiency as the ratio of diffracted energy to the incident power to see how much light we're losing due to diffraction. So this is an issue in candle because when we shape the spectra, we want to make sure that there's enough light that we're sending out in the infrared regime. So my project is testing, how, if we use a different DMD pitch, for a certain wavelength regime, can we maximize efficiency in comparison to another wavelength regime? So I tested these two DMDs. And so the experimental setup is essentially to focus light onto this DMD and then measure how much light is diffracted off of this DMD. And then we replace this with a flat mirror. Oh, that ray looks off. But we replace the DMD with a flat mirror angled similarly so we can take a measurement of the incident power. And this provides us two spectra that we can then divide to get the diffraction efficiency. So first of all, when we do these for the two different DMDs, we notice that there are peaks in efficiency, which is what we expect from the Blaise diffraction grading model. And also, it actually matches the theoretical expected Blaise wavelength peaks. So you can see that from the orange dots, which are the expected locations of the peaks in efficiency. So this is good, but we care mostly about comparing these two DMDs and seeing where the efficiency is maximized for either one of them. So specifically, you can see for the DLP 650, which has a smaller pitch, the efficiency seems to be maximized between 1400 and 1800 nanometers, whereas for the DLP 7000, it's maximized between the 1600 and 2400. And so this maybe allows us to adjust the wavelength bands that we are working in for candle. So ultimately, this will help us inform some design choices about candle. And not only this, DMDs are really interesting and useful, like beyond just candle, and they're applied in different classroom projectors or hyperspectral imaging and atom trapping experiments. So they're a very versatile device that are really interesting to explore. Thank you to my mentors, Tin, Susanna, and Joe for mentoring me throughout this project, and also NIST for this opportunity, and also ASF for all of this. Thank you.
Thank you, Brianna. Question.
Where are we in actually using this to improve our ability to measure the distance to the universe?
That's kind of a tricky question because I guess the goal of this is to provide improved flux for these type 1 supernova measurements. There's actually an issue called Hubble's tension right now where these two different methods of measuring the Hubble's constant or the expansion of the universe are conflicting, and it's a big problem in cosmology right now. This will help constrain one of the methods, but it cannot account for the other method's difference. So hopefully by constraining the Hubble's constant version, the type 1a supernova version, we can get a better assessment of how far it is from the other measurement. Yeah, thank you.
One more question. Anybody?
Very interesting talk, thank you. Since you're at NIST, do you have any comparison of using this versus using frequency combs for spectral data calibration?
No, I do not, sorry.
Okay.
Short question. All right, thank you so much, Brianna. Next, we have Kaylin Smith analyzing the impact of performative and adapted autonomy on collaborative robots. Maybe they're swimming with the fish, we don't know. Welcome, Kaylin.
All right, good afternoon, everyone. I'm Kaylin Smith, and that is not my presentation.
Can we?
Sorry.
Yeah, we'll switch it up just a second.
Yes, that's great.
There we go.
Thank you. All right. I'm Kaylin Smith. I'm going to be presenting my research on performative and adaptive autonomy for collaborative robots, which I conducted at the Mirror Lab at Colorado School of Mines. So I want you to imagine that in the future, you're an engineer at NASA, and you're working with some new Mars rover. And this rover is asking you what it should be doing. While you're deciding that, you're also handling a lot of other tasks. Maybe you're analyzing some data, helping an intern who's really confused, and writing a new business proposal. And on top of all of that, that robot is still asking you what it should be doing, even though it's capable of deciding for itself. Now, on the one hand, this might be really frustrating, as you have all these tasks going on, and you don't want to be helping that robot on top of everything else when it could make that decision for itself. But on the other hand, this might be helpful, as you likely weren't paying attention to the robot, and so it asking you this might force you to pay attention to it, and that will help if you have collaborative tasks with the robot, or if you need to take over from it if anything goes wrong. And so this is really the question that we're trying to answer. How autonomous should a robot be in order to increase the awareness you have of what it's doing without overloading you with information, specifically in settings with communication latency, such as a robot being on Mars? Specifically, our study is evaluating the impacts of performative autonomy in settings with communication latency. Performative autonomy is a design strategy in which robots intentionally lower the autonomy that they're using compared to what they're truly capable of in order to increase the situation awareness of a human collaborator without posing a substantial increase to their cognitive load. So in the example of the robot on Mars, it asks you what it should do, and then you likely will pay more attention to it, hopefully without burdening you with information. And we aim to investigate the effects of performative autonomy in settings with latency on cognitive load, situation awareness, and key metrics for human-robot teaming, including trust in the robot and your perception of its intelligence. To evaluate these effects, we conducted a human subjects experiment with 76 participants. In this experiment, participants played a resource management game with a robot in which their continued progress required certain resources to be collected by a remote robotic partner. They could see a live stream video feed of a remote robot, which was this Misty 2 robot, and latency was imposed as a two-way communication delay between the human participant and the robot. We manipulated two independent variables: the amount of communication delay between the human and the robot, and the performative autonomy strategy used by the robot. Each of these had 3 levels, and the 3 levels for performative autonomy strategy were based on how the robot communicated with the human. Whether it asked the human to make all of its decisions for it, whether it made a decision but required human confirmation, or whether it made all of its decisions without any human input. Following the experiment, we conducted statistical analysis on the results. Contrary to prior experiments, we found that lower autonomy caused an increased cognitive load with no effect on situation awareness. And we believe this was likely because our experiment imposed a higher baseline cognitive load than prior experiments, which shows that performative autonomy might not be effective if the human is already under substantial cognitive load. Secondly, we found that higher latency caused a decrease in trust and decrease in perceived intelligence of the robot. And this shows that robots might be implicitly blamed for latency, even though that's solely due to the fact that they're in a remote location. And that then must be accounted for when designing remote collaborative robots. Lastly, we found that the level of autonomy of the robot had no impact on trust or perceived intelligence of the robot. And this is important and encouraging because it shows that performative autonomy might still be effective in maintaining good human-robot teaming. Although there's many future directions that we could take this work, what we're currently doing is exploring the impacts of dynamically changing the robot's level of autonomy. And to do this, we're developing a deep reinforcement learning algorithm that dynamically changes the robot's level of autonomy in real time based on the human's cognitive state. So as we expect that robots will need to collaborate more and more with humans in the future, including in remote locations, this study showed some impacts of performative autonomy and perceptions of the robot that are based on its remote location. And I want to thank Dr. Tom Williams and everyone else at the Mir Lab for supporting my research, as well as to the Astronaut Scholarship Foundation for giving me the chance to present my research here. Thank you, and I'd be happy to take any questions.
Thank you so much. Let's have a question.
Hi, super interesting talk, thank you. So I was just wondering, the very last bit that you mentioned about how it would change how it was acting based on the human's cognitive state. That's very broad. I was wondering if you could talk a little bit more about if there's any certain features or what that would look like.
Yeah, that's a great question. And so we're tying this into the work I talked about very closely. So we'd mainly be measuring cognitive load and situation awareness in real time and be adapting it based on those factors that we're measuring from the human.
Thank you.
Another question.
Hi. In your Mars example, you mentioned how mission control will be working on training interns or doing other secondary tasks. Is there any ideas into doing research with this where your subjects in, say, this paper are doing other secondary tasks as well, the multitasking aspect, if that increases the cognitive load and how that affects their views on the robot's autonomy?
Yeah, that's a good question. So in the experiment that we run here, they are doing quite a few other tasks, and that's one of the reasons why their cognitive load is very high at a baseline in this experiment.
Gotcha,
Yeah.
thank you.
I have a question.
Yeah.
Can a robot be told from a situational awareness perspective to unload the dishwasher?
I wish.
I
I think
wish
we need
too.
some of those robots.
Let's work on that, okay?
Yeah.
All right, that was fantastic. Thank you, Kaylin. We'd like to welcome our next presenter, Louisa Mao. She's going to be talking about semantic masking and visual feature matching for robust localization. Welcome, Louisa.
Hello. Oh, wow. Hi, my name is Louisa Mao, and I'm presenting over some work that I did with NASA Ames called Semantic Masking Visual Feature Matching for Robust Localization. And what all of these very long words mean is that we're just trying to make localization better for robots. And in robotics, localization means exactly what it sounds like it does. Basically, we're trying to find the robot's pose in the world given the robot's sensor observation of the world. And if your world happens to have 3 dimensions, then a pose is an x, y, z coordinate plus an orientation. And in visual localization, we're working with images. So when we get the camera sensor observation as an image, we first run an algorithm over it to detect key points, which are just like distinctive groups of pixels. And we also have a map, which also has key points, except we've already found the 3D points for each of the key points in the map. And having this, we just need to find the correspondences between the points in the 2D image and the points in the map. So then we can do some fancy linear algebra calculations to find where the camera needs to be viewing from such that it can see these 3D points projected onto these 2D pixel locations. And we want to do this localization thing for robots on the International Space Station. And this was supposed to be a video, but it's not moving, so you can just use your imaginations. As you can see, this is a very visually busy scene, which means localization is really hard because all of these wires and everything are generating a lot of key points. And these key points can't even be used because everything is floating around in microgravity, it's not stable, maybe astronauts are rearranging things. And we can't remap this environment frequently enough to capture all of the changes that can happen aboard it. So really, we're depending on the localization algorithm to be robust enough to handle these changes. Now, this is the actual algorithm that's running on the Astronaut robots on the ISS. First, we've got the image that the robot is seeing, extracting the key points, and then we use it to pull up similar images from the map that are hopefully of the same place where the robot is looking. And then we do this feature matching between the 2D points in the image and the 3D points in the map. And then once we have this correspondence, we can do this linear algebra and then find the robot's pose. However, in this feature matching step, there is a lot of potential for errors to be introduced because, first of all, the objects on the ISS can move around and then you can't use those to find the robot's relative pose. Or you can just get false matches, because maybe you could get a key point in a white wall that's matched to a light, just because those pixels look kind of similar. So in our approach, which is shown in the blue boxes, instead of just relying on these tiny little key points, which can be pretty inaccurate, we can actually leverage this object level visual information to help us out with localization. And so at the same time as we are extracting the key points from the images, we're also running an object detector neural network over the images, which tell us where the lights are in the scene, or the vents, or the handrails, or those kind of larger objects. And then when we do the matching step, we only match the features that are within the same object type together. So in machine learning, we call this like semantic classes. This is like when features in lights are matched only to features in lights, and then vents to vents, and so on. So we're essentially filtering out all the incorrect matches that can happen, and this gives us a better pose calculation. So I'm not going to show all the boring quantitative results, but this is a really cool qualitative example of an actual deployment that was on the ISS. So what's happening here is the robot is doing a yaw angle rotation. It's spinning around the z-axis. So it starts off facing one end of the Kibo module, and then it spins around and it looks at the walls, and then it faces the other end. So on the left side is the baseline performance, and this image pair comes from one point in the middle of the trajectory. And then the chart under that shows the, shows the yaw over time. So intuitively, you should see the yaw angle smoothly increasing over the time as the robot is turning. However, we see this weird little discontinuity here. And what's happening is that in between creating this map and using this map to localize, somebody, I don't know who, turned this flag upside down. So when the robot is looking at this flag, it thinks it's upside down. And you can see how this angle is reversed, and it thinks it's turning around backwards. So on the right side, in our approach, you can see that the incorrect matches here in the middle in the flag get filtered out. So when we calculate the pose, we get what you would expect to see, and this kind of, mildly catastrophic situation doesn't happen. So in conclusion, we made a computationally efficient improvement to the NASA Astrobee localization system, and it is robust to changes in the deployment environment. I'm happy to take any questions.
Thank you. Who has a question? All right.
Thank you. I did have one question. Could you use your algorithm to do just the opposite and make it more difficult for something to localize itself? In other words, to optimize camouflage?
Oh, well, I see. Okay. In that case, you would want the opposing side to implement an algorithm that makes it more difficult for them to find you, I guess. I'm not sure how you would convince them to do that, but if you could, then I suppose so.
Another question? Any other questions? Wonderful, great presentation, thank you.
Thank you.
Let's now welcome Kavi Day for fMRI encoder alignment image reconstructions from brain activity. Welcome, Kavi.
Hello, everybody. I'm Kavi Day, and I'm going to be talking about fMRI encoder alignment. I don't want to talk about the boring title, so let's jump into a question for all of you, which is what do Professor X and cutting-edge machine learning have in common? Rhetorical question, there's a ton of answers. The one I want to focus on is mind reading. And I want to be really specific what I mean by that. If you look at an image while we're scanning your brain, we can take that brain scan and use the brain scan to generate the image that you're looking at. So aside from being really cool, what are some practical applications of this? One is an increased understanding of the human vision system. Another big one is an accessibility tool. It's another way of communicating, right? If we can read brains and we can understand what brains are thinking. And I also want to point out that this is a non-invasive technique. I'll highlight exactly how that works on the next slide. So this is going to be a crash course in mind reading. Please ask me questions later if you have some. I also want to point out my background is not in neuroscience, so feel free to correct me on anything. But we start with a human participant who's in an MRI machine. While they're in this MRI machine, they're looking at a visual stimulus, in this case an image, and we're recording fMRI bold signals. So bold signals are basically a measure of blood oxygenation, which is a proxy for brain cell activity. So that allows us to see which parts of the brain are active, which neurons are firing, what is a person thinking at a high level. So we collect these brain signals, which are from the scene image, and then we need to use them to generate the image the person saw. Most of you are probably familiar with ChatGPT, and you may have even used it to generate images before. So let's say you went to ChatGPT and you asked it, generate me an image of a cat eating a hamburger on a skateboard, for example. Some people like to do that stuff. So it uses what's called a diffusion model, which can take a text prompt or an image description and then turn that into an image. I'm not going to talk about how diffusion models work, but the most important thing you need to know is it can take any image description and turn it into an image. So our challenge is then, how do we take a scan of somebody's brain and turn it into an image description that can be used by the diffusion model? We do that using a model that we call a brain encoder. And for those of you who are machine learning nerds, we technically use an autoencoder with some transformers, but the details aren't super important. So we start with a brain signal, we generate that, we turn that brain signal into a brain description, and then we take that image description, which we call an fMRI latent, and then give it to a diffusion model, which turns it into an image. Super high level pipeline. Please ask me if you have questions. And there's obviously a few more details when you actually need to implement this in code. So to go back to my original statement, what do Professor X and machine learning not have in common? One of the big ones here is that this is not portable. MRI machines are the size of rooms, and it requires consent, right? The government isn't like pointing a machine at your face and seeing what you're thinking. And that consent part is really important. It actually requires 30, 40 hours of data per person before you can read their brain. So if I train an algorithm on you, it cannot be used on anybody else. So it really needs to be fine-tuned on each person. And because this is so expensive and so hard, there's a lot of limited data, which is never good when doing machine learning. And that means that the results, while I think very good, often have pretty low quality images. So on the right side here, you can see the ground truth image the person saw, and then the image that our algorithm generated. And I want you to focus on the far right image. So at the top, there's a person swinging a bat, and the bottom, there's what might be a person maybe playing a sport. I don't know, like the legs are in the wrong spot, and if you look at the bug, it's kind of like malshapen. So the focus of my research is how can we take these generated results and improve them and to make them look like actual images that aren't out of some kind of nightmare. So how can we increase quality? Remember that we're using an existing image generator. So our goal is not to touch the image generator. We want to make the best brain encoder possible while leaving the image generator alone. That means that we need to give some kind of good feedback to the image generator, and there's two kinds of feedback that you can give. The first kind is direct feedback. So the way this works is the image generator gets the image encoder gets the brain encoder gets a brain, and then it gives us an image description. And we can look at that image description and say, this is a bad description, this is a good description, and try to evaluate it. But deciding what a good image description means is pretty hard because an image description is a vector in some random latent space. So it's not a thing that you can really talk about. We're much better at describing whether an image is a good image or whether an image is a bad image. So my research is about how we can take the image description that we generate, give it to the image generator, then look at the image that was generated, and we compare the distribution of all the fake images that we generate with the distribution of all the real images that a person actually saw. And by trying to align the two distributions, you can basically improve the fMRI encoder. This is like a super high-level overview, and a lot of these techniques come from the area of distilling diffusion models, specifically score distillation sampling and distribution matching distillation. Again, please ask me if you have questions. It's a bit beyond what I can talk about here. But these are some preliminary results. You'll notice on the left side, we have the ground truth and the original reconstruction before we applied my technique. And on the right side, you have the ground truth and the improved reconstruction after we applied my technique. If you look at that horse-shaped blob that has no detail, that's what we try to turn into a nice-looking, if you look at the middle example, like a house on a lake that has good reflections and it's a much higher quality image. There's a few areas of future work. So right now we've done images, but we want to do audio, video, 3D models, et cetera. We also want to understand more about how these algorithms actually work. You can see an example of understanding how different layers of the model pay attention to different parts of the brain. I want to thank NUS and also ASF for allowing me to give this presentation, and this slide is just here to help you ask some questions about my research.
Thank you. All right, lots of questions about reading minds.
Hey, awesome talk. I wish I knew more about machine learning, but why not go straight from the fMRI data to an image? Why two models?
So training a model to generate images is really, really hard and it requires a ton of data. The data set that most image generators are trained on is called Lion 5B because it has 5 billion image text pairs. If I had 5 billion brain image pairs, I would be the happiest researcher alive. But unfortunately, we don't have that, so we have to do this roundabout technique.
All right, got it. Thank you.
Another mind reading question.
Thanks. I'm wondering whether you have found any significant difference about female and male brains, that the mechanism, how they generate images or signals.
So we have not. The specific data set that we use in this paper has, I think, 6 participants, maybe 7 participants total, because it's so time-consuming to collect 40 hours of data across a whole year for a single person. And in most cases, each person doesn't even come to all of the sessions for data collection. So you have limited data from different people. So there isn't enough research into this and enough data yet to make those kind of conclusions, unfortunately.
All right, well, thank you so much, Kagi. I love the part about the government doesn't want to be reading brains. Tiernan, Dotley, blocked versus random speeds. Which structure is better for learning catching skills in VR? Welcome.
Thank you. My name is Tiernan Dottle. I'm a computer engineering and math major from Louisiana State University. I'm about to take us into a different realm because I'm here to talk to you about football, specifically a catching skill. And it's very different because I research for the LSU School of Kinesiology. So today we're just going to talk a little bit about my topic, how we went about this, the procedure results, and some future directions for this research. So VR, as you all know, is becoming increasingly prevalent in our lives. If you think about systems like the Oculus or the golf simulators, it's becoming more and more prevalent. And on the research side, there has been some research done using VR. Unfortunately, there's been a variety of research, so it's led to weak precedent in this area. So the major advantage of using VR training as opposed to training a catching skill in real time would be that we can manipulate the speed, making it either faster or slower that the participant sees this throw. So speed manipulation is important because in past research, we've seen that it has some benefits, including reducing the amount of training time required and also better task transference, which is when you teach a skill in a simulation and then you move it to doing the actual task. However, most programs still don't include any form of speed manipulation. So another important part of teaching a skill is how you generate the training. And so we look at two basic forms, which is random versus block training. So if you take all of the different training throws for us, and you break them down into smaller subgroups, these are what we call blocks. And in block training, there is no variation within the block itself. And when random training, you have variation within the block. There is differences between blocks, though, in terms of speed, which is what we look at. So the purpose of our research is to test the effectiveness of random speed training in VR. And our research question was, will blocked or random training elicit more improvements in VR football catching? And we believed, based on the literature, that the group with block speed training would perform better throughout the training, but the group with random speed training would perform better at the retention test. So this is a protocol. I'm just going to breeze through it for the purpose. We had 41 college-age participants. They came in and did a trial, a pretest with 12 throws, and then we broke them into two groups, 6 blocks of 12 throws each for a total of 72 throws. And then the next day, they came back 24 hours later and performed the same pretest. So because we're talking about VR, I thought we would get a visual. On the left-hand side of the screen is what the participant sees when they're playing the game. There's a quarterback that throws the ball to you. And on the right-hand side is one of the participant views, so how I can interact with the participant while they're in the setting. So our variables of interest were trajectory percentage, the blocks, we used 6 training blocks in this study, the speed, which is we did random versus blocks, and our dependent variables were catching percentage, visual reaction time, hand reaction time, and movement time, all metrics that have to do with how fast you react. So this is our results slide, which is a little bit busy, so we have summarized it for you. Overall, both groups showed significant but similar improvement, which is not what we were expecting at retention, and there were no changes in response behavior. In catching specifically, there was performance decrease with random training compared to block, which is what we expect because it's harder. But there were no benefits at retention, and looking at the literature after this, we noticed that in some non-laboratory trained tasks, this was very similar to the results that they saw. In response behavior, we saw that there was sensitivity to different speeds and difficulties, so how much they learned depends on how hard and how long they trained, but there were no permanent changes that we could elicit. In the future, we would love to try this with different sports tasks. This specific task was based on an LSU football team drill. But we would love to try other sports and different tasks because the difference in tasks can sometimes relate to how well people learn the skills. We would like to implement new VR technologies like haptic feedback, playing with other people, or wireless play because the headset we use has a really long cable that we hope that no one trips over while playing this game. And we also want to do transfer tests. That's a really important part of simulator training, is showing that it has benefits in when you do the task itself. And we were unable to get time at LSU football to try this out with our participants. So that's something we look forward to trying in the future. I'd like to thank you for your attention and thank the ASF for including me in these presentations. I would be happy to take questions at this time.
Thank you, Tiernan.
Quick question. So, was there a way to test, so traditional old school football drills on the field with a coach or a trainer? Were you able to do sort of, again, compare and contrast, those skills or, improvements in efficiency and catching versus the VR environment? Were you able to test the difference between those two and the efficacy of each?
So that's the transfer task that we wanted to do. Originally, that's how we were supposed to do it. We got the LSU football players to do one session of our training, then we were supposed to compare it. But getting time with the team is kind of a hard thing to do, even during spring session training, but that is definitely something we want to do. The other difficulty with this is that we use a ball in the game that's 40, 43, and 46 miles per hour. These are speeds that you probably wouldn't want thrown at your face when you're 10 yards from it, because that's how this one went. So with our college-age participants, it's kind of hard to put them out on the field and say, don't get hit in the face by a football, but try and catch these.
There's also an online question that I would like to have asked. So they asked, were the participants able to move around much during the test?
So specifically for this task, we have them plant their feet and get in an athletic stance. They were told that the ball would be thrown like a very good quarterback to them, and they would not have to move their feet at all. We had some people who had happy feet and would like to, get away from that starting position, but there's a green target on the ground to make sure that they're always starting at the right position. But yes, with wireless play, that is something that we would like to implement. There are different drills that involve basically running a route and then catching from something like that. So that is something we would love to look into.
Great, thank you. It's almost football season. Thank you, Tiernan.
It is. Go Tigers.
All right, we would like to now welcome Clara Hung on parallel dataset acquisition for lensless imagers. Welcome, Clara.
Hi everyone, can you hear me okay? I know it's the afternoon slump, so this is going to be super high level. If you have any questions about the technical details, feel free to come talk to me after the talk. But I'm Clara. I'm a senior at UC Berkeley studying computer science and physics, and I'm going to talk to you about some of the exciting things I've been working on in our computational imaging lab to advance a field called lensless imaging. So a lot of the work that we've seen today relies on imaging in some form or another. And with traditional imaging systems that we have, we're kind of reaching the physical limit of what the physics and the hardware of these systems can provide, whether that's microscopes, cameras, or telescopes. And a big question in the field of computational imaging is, how do we see more with less? So can we try to dial down on the hardware and leverage new developments in, say, algorithms and machine learning to see more with less? So this is Patrick, by the way, Patrick the penguin. The imaging systems we usually see in the lab today have a lens and a sensor for us to see Patrick. And for more imaging power, that lens can get really bulky and expensive. So here's a question. What if we just remove the lens? What if we removed the lens and instead used an optical element? In our case, we use a face mask to replace that lens. Can we still image something with this system? And it turns out we can, but at first, the measurement looks like garbage. So what is happening here is that the face mask encodes information about Patrick in our encoded measurement, and then we send that through a reconstruction algorithm to decode that measurement into our final reconstructed image. So this is Patrick on the other end right here, and this whole process is our lensless imaging pipeline. Now, lensless imagers currently aren't very widely used in practice because the quality is still not on par with that of our lensed imagers. There's a reason why we have been using the same lens and sensor system for a long time. But we're still working on improving the image quality. So nevertheless, lensless imagers do provide a couple of advantages over our lens systems. So for example, it is super compact. Lensless systems with just a simple face mask and a sensor can be on the order of millimeters. In addition, the face masks that we use tend to be a lot more inexpensive compared to their lens counterparts. And with compared to a similarly designed lens system, we can actually get a wider field of view with our lensless system. And building off of what Kavi was saying, a lot of the work we're doing nowadays is really data-driven, and so is lensless imaging. And data sets are often the bottleneck, as we saw in your work earlier. So everything from designing the phase mask, trying to see, how do we learn the best phase mask that improves image quality, to investigating different machine learning-based reconstruction algorithms, or even end-to-end system design. So that's improving a little bit on the hardware and the software over many, many sets of data to see if we can get the best system. All of these areas of research require a lot of data, and currently we do have simulated data sets that are at the scale that we would like, but simulated data sets aren't able to capture the non-idealities in the physical imaging conditions, which may result in model mismatch later down the pipeline. So ideally, we'd like to have really large experimental data sets, but currently the data sets in the field don't quite meet those demands. So what I'm working on in my lab is a way for us to acquire large experimental data sets with multiple different lensless imaging systems at the same time. So the idea is that we can scale it up so that if we wanted to acquire from 3 to 4 different systems at the same time, we can get measurements from different architectures and compare them under the same imaging conditions. Oops. Another important piece is that we have a lensed camera in our system, and that provides a ground truth camera, which is really useful in machine learning applications. So with this data set acquisition system, we're able to capture the large-scale experimental data sets that will really facilitate a lot of emerging work in the field of lensless imaging. And at the current moment, we're still fine-tuning our hardware system, but we have a proof of concept, and I'm really excited to get the full pipeline running in the coming year. And to wrap it up, I'd just like to thank the Computational Imaging Lab, especially my PI, Laura Waller, and my mentor, Leila Kabouli, and everyone in the ASF family for allowing me to present today. Thank you.
Thank you.
That was super interesting. What is the, after image reconstruction, what is the resolution that y'all are trying to achieve?
This is a good question. Let me show you the pipeline. But it really just depends on the system or the lensless architecture that you're using. So different lensless cameras have different resolutions, but currently we're trying to compare with our ground truth system. Yeah.
One more question.
Do you know, this is on the same idea of resolution, is there a theoretical maximum resolution you can get from a lensless camera compared to like a one with a lens?
Ooh, that's a good question. So when we primarily calculate resolution, we've been using like the 2-point, or our lab actually developed a 16-point resolution test to test like how much we can achieve, but lensless systems have been able to achieve super resolution, so there's still a lot of work going on in that though.
All right, thank you so much, Clara. And last but not least, we have Ryan Rodriguez, on-demand multimodal transit systems, physical and computational case studies in 4 U.S. cities. Welcome, Ryan.
All right, thank you. Oh, there we go. That's great. All right, so hello everyone. My name is Ryan Rodriguez. I am a research assistant in Georgia Tech's NSF AI Institute for Optimization called AI. Whoop. How do I go back on this thing? There we go, yeah. So our work focuses on public transportation systems, and what's really unique and interesting about our lab is that we have the ability to conceptualize these systems, then simulate them, and most importantly, actually put them into practice with real-world deployment. So the system that I'm about to show you here today, on-demand multimodal transit systems, we abbreviate it ODMTS, I'll talk a little bit more about it later, but we actually had the opportunity to deploy this in a couple of case study cities. So first off, let's talk more broadly about public transit. And public transit has a problem, at least for the United States. Public transit utilization in the United States is very low. Actually, about only 1 in every 20 people regularly uses public transit to commute to work or school. And a lot of this is due to how our public transportation systems are structured. Currently in the United States, a lot of public transportation systems are very slow. They have imperfect transfers. You can see here for a sample route in the city of Atlanta, where GT is located. Just to do a route that would take typically 15 minutes for a car, you'd have to take, I think, 3 or 4 transfers, which is very inconvenient. And then also there's a first and last mile component to that as well. So you aren't able to be serviced directly from your origin to your destination point. There's always a walking leg or a driving leg to get to the system or get from the system, right? So fundamentally, though, we know that public transportation is socially good, right? It reduces congestion, it reduces emissions, it's often cheaper for users, and also, last or most importantly, you don't have to pay for parking. Nobody likes paying for parking. So in order to improve our systems, we have to address some of these key inefficiencies. So whatever solution we create, we want this solution to be fast. We need this to have synchronized, more simple transfers for users to undertake. And then also, most importantly, we need to have first and last mile service. This means that you can walk outside, press some buttons on an app, have a vehicle pick you up from your doorstep and get you to your destination without having an initial walking or driving leg, right? And of course, ideally, it would be nice to have a solution that's affordable for both users and transportation agencies, and then also that has minimal disruptions to existing core services. And what this means is that if you're accustomed to taking a bus leg to work every day, we want to keep those intact as much as possible, right? All right, so enter on-demand multimodal transportation systems. The on-demand portion means that the transit systems are driven by the users, right? So when users want to take a trip, they'll open up an app, they'll input in their origin, they'll input in their destination, and the multimodal means that there's multiple modes in order to get from your origin to your destination, right? So for the key systems that we're seeing here, typically we'll have a shuttle. You can think about those as like short Ubers that make short hops for short distances. Bus rapid transit, just bus legs, and then also heavy rail and light rail as well, which you can think of as like metros or subways, right? And these systems work together to bring you from your exact origin to your exact destination there. You might have a shuttle leg, and then a rail leg with a bus leg, and then finally that brings you to your car. And at each leg of the journey, the next mode of transportation is waiting right there for you. So even if you're doing 2 or 3 transfers, there's no waiting time, there's no walking or driving time in between. It's very simple, right? You can see a map over there on the right of showing a simulated pilot in Atlanta, right? So great. So these systems work conceptually, right? But now we need to simulate them and see if they work here too. So this is a quick pipeline here. I won't go into the details as much for this, but the main idea is that we have transit information on these origin-destination pairs that I talked about earlier. We create an initial simulation in the ODMTS design portion right there. I'm going to show you a little bit about those in a bit. And then finally, we'll simulate those to get a final simulation. So the data that I'm about to show you for 3 computational case studies is just an initial data before those ride-sharing fleet optimization portions in purple that we saw on the previous slide, right? So these systems and these numbers can be improved in the future significantly. So for a small-sized city, we can see significant cost and time improvements. For a medium-sized city, we can see those same improvements while still keeping a lot of the existing bus routes that you can see there. And then also for a larger city like Atlanta, we continue to see those cost and time improvements. Well, also, one important thing as well, keeping drivers employed, existing drivers, and minimal switches between vehicles. So typically, we like to reuse the same vehicles, right? So not only conceptually, but also for simulations, these systems work too. But now, does it actually work in practice? That's the real question, right? And the answer is that it does. So there's 2 cities that we've actually done real-world deployments for this system for, right? We've actually bought the shuttles, we've hired the drivers, and we've made these systems work and available to the general public. The first example here is with Atlanta. We got $1.2 million from the National Science Foundation to conduct a real-world pilot for 6 months. Everything is developed in-house. So that means that we get approvals from the transportation agency, MARTA, but all the dispatching, all of the troubleshooting, that all happens within the lab, right? And we saw some significant ridership improvements there on the left, and then also customer satisfaction was through the roof compared to existing MARTA service, which typically is quite good. And then also, This pilot is still ongoing in Savannah, Georgia, but we've seen those same cost and time improvements as well as user satisfaction and ridership increases as well, right? So across the board, in conclusion, on-demand multimodal transportation systems work both conceptually in simulation and then also in practice. I'd like to thank the AI Institute for Optimization, the National Science Foundation, and the Astronaut Scholarship Foundation for supporting this research. I'd like to take any questions. Thank you.
Thank you, Ryan.
Hi, great presentation, first of all. But I was wondering how your system is able to handle so many users, because I know apps like Uber often get overwhelmed with the amount of people trying to get there, especially during rush hour. How does your system combat that?
That's a great question. I do want to start off first by saying that the pilots, while these are large pilots, probably some of the largest that we've seen out there for these types of systems, no transit agency will let us immediately take over all of their public transit system operations for a city of around like 6 or 8 million people, right? Even though we haven't seen as many users now, I will say that the dispatching systems and things like that are very robust to a lot of requests. So we would be able to handle larger volumes if we were given the time and finances to prepare.
Okay, thank you.
Another question?
Great talk. I wanted to ask how your system would factor in traffic and how that would affect the wait times in between transfers.
That's a really good question. So first off, this system actually reduces traffic. I know it may seem a little bit counterintuitive with us bringing in shuttles and buses, but it does function like a regular transportation system, so it does reduce traffic. In terms of transfers, what is really nice is we incorporate ride sharing, right? So what might seem like a 1 or 2 minute detour to one person can drastically reduce the waiting time for somebody else, right? And then also typically we like to have more shuttles than we need for certain periods of demand. So there might be 1 or 2 shuttles that are just latent, they're just waiting for new users to come. Yep. Thank you.
Fantastic. Thank you, Ryan. Nobody wants to wait for a bus, especially me. So come to Minneapolis, okay? All right. Thank you so much, everybody. I'd like to have one huge round of applause for our 18 afternoon scholars. Fantastic. So I want to thank you all. So my dad is, I think he's watching probably right now. He's going to be 87. He's fascinated by everything going on here. Madeline knows my dad. And he's been texting me the whole time because he's really good at texting, as is my mother, but that's a different story. However, all I got was, wow, amazing, incredible, the future is bright. So yay, fantastic. So on that note, Carolyn's going to close us off with some logistics for tomorrow. But I want to say the world awaits all of your brilliance. Have a great rest of the night. Thank you.
And a big round of applause for our moderators for keeping us on time. Are you up for the challenge tomorrow as well? All right, great job, scholars. These were excellent presentations. Thank you so much. And thank you to this incredible audience. You were very engaged. You asked great questions, and thank you for asking the questions because I would not have known what to ask. So good job. Scholars, turn in your scavenger hunt sheets to Janelle or Lisa by noon tomorrow. And session 3 for the Astronaut Scholar Technical Conference starts at 9 a.m. sharp back here. Got that? All right, everybody have a great evening. Thank you.